we're hitting 5,000 transplants, and they want to celebrate.
Okay.
And so September, then it was going to be the 21st, but now they're telling me September 28th. But they haven't yet. They're going to confirm by Monday next week. So, if that case, 16 to 20 would work.
Okay, let me know then.
Yeah, I knew we will. But thanks, Jenny was reminding and clarifying that.
Otherwise, we can change the dates. Yeah.
We'd share what?
Huh? Otherwise, if you are not available, we can change the dates.
Oh yeah, right. No, that's what we were just talking about. But again, it was when she asked me about the 16th to 20th. The 21st is when we they were planning, but it looks like they changed it to the 28th. So the 16 to 20 right now would work.
Okay.
But they said they'll give me a definite answer on Monday. So hopefully by the beginning of next week, I can,
Yeah.
Jenny, I know Jenny will remind me. So
definitely I will do that.
Richard, I have one request. One of my students who is now working in Phuket, in the south of Thailand.
Yep.
She would like to spend some months with you, similar to the previous one, next year. So I will let you know. I will write you an email then.
Okay. Sure. It's working out very well for Dr. Pangapan. I think she's really enjoying and learning from the team. She's certainly seeing a lot. I think we're looking forward to Smith coming and visiting. And yeah, I think we can work with that. Is that person from Phuket?
Yeah.
Is that someone that is a transplanter now or wants to be a transplanter or?
No, she will spend about a year at Sirat Hospital working in the transplant unit before going to IS.
Okay. No, that'd be great. We'll look forward to accommodating.
Good.
And
I'll write you an email then.
Good. And I have to send you an email about a very potential interesting opportunity.
Yes.
So for where, if we could set up a collaboration on CAR-T between where patients from Thailand can come here and then get all their follow-up over there, but it'll be on a clinical trial with Miltenyi, and so all Miltenyi will pay for is the making the product, but we've got to figure out a way that people can fund their care, for when they come here for 4 weeks, and everything else would be over there. But we talked about this in Europe. They're very excited about trying to make this work with you, and I said I would get back to you. But since you're on the phone, I'll just streamline that conversation that you and I will be emailing about that.
Okay, good. Sounds very interesting.
Yeah, no, it's a good opportunity. I think it could work. It'd be very interesting. It would connect our institutions really tightly.
Mm-hmm. Good.
Agree, Dr. Richard, agree.
Yeah.
So I just said agree with what you and Dr. Surapon just talked on the joint research on CAR-T together. Today you're going to talk about the oncolytic virotherapy, right?
Oncolytic Virotherapy, yeah. So if you want to hear, I can start.
Yes. Please.
All right. So this is something I'm sort of interested in a little bit. I'm going to just put it on slideshow. There it goes. And hopefully you can see that. This is a talk I've never done before, but this is something that's becoming very interesting. And although it's not a reality right now, it's something that I think within the next 5 years, we're going to be able to be combining oncolytic virotherapy with CAR-T therapy. So what I thought is maybe it's time for us to learn about oncolytic viruses. And so, of course, you have to understand viruses. And by the way, this is the SARS-CoV virus. That virus has RNA or DNA genetic material, has a protein coat, and often has other molecules that allow it to adhere to a surface. So the outline here is going to be, we're going to review the life cycle, briefly life cycle of how viruses exist, what the rationale for capturing, viruses as a therapeutic tool, talk about some human work that's happened, clinical trials, whether it's best as single or multi-agent therapy, and talk about what's out there now in America. There's 2 viruses approved. There's some approved elsewhere. And then how this is going to lead to future combination therapy. So first is some very basics. Now, I learned all this stuff but many years ago, and I just know there's viruses. I know I deal with CMV, and I know that's the double-stranded DNA virus, and I know that I give people vaccines for measles, mumps, rubella. I give vaccines for, HPV, but for the life of me, I forgot a lot of the basic biology. And so going back to David Baltimore got the Nobel Prize for reverse transcriptase, but what he's created years ago, this is the Baltimore virus classification system, that there are 7 different classes, because you have double-stranded DNA virus, you have single-stranded DNA, that's a positive or the negative strand, you have double-stranded RNA, you have negative strand RNA, a positive strand RNA, and what's shown in that little bottom is these are all the 7 classifications, what they might look like when you look at their DNA or their RNA. But what the whole goal is, the virus will survive only if and only if that particular genetic material is transcribed into mRNA in a eukaryotic cell that ends up making itself and replicates itself. So we look at more basic biology. In single-stranded DNA, sometimes this will actually requires the cellular DNA polymerase to make a double-stranded DNA that ultimately will split to make more single-stranded DNAs, but it also has to have the cellular RNA polymerase to transcribe into RNA and to make new protein that will then ultimately make the nuclear material for the signal, as well as the protein that goes into the capsid. And so, but it's a simple material, but it allows itself to replicate quickly. Double-stranded DNA, viruses often have a step where it has to get into the nucleus and use nuclear enzymes to replicate itself, as well as cytoplasmic enzymes to make the RNA. And so there is different type of viruses, and these different viruses, some will code their own polymerase, some will actually use the host cell polymerase. And so it's a whole complex set of biologies, but again, if you could break all the viruses out there, you could break it into 7 different classes. This is one last picture showing, again, a little bit more about the DNA polymerase, into the reverse transcriptase, the RNA polymerase. Key is making protein that will ultimately lead to making a capsule and envelope. Once you have a viral particle, it gets into the host cell. Again, it can use nuclear material, it can use cytoplasmic material, but it has to replicate itself, generate all the proteins that will allow it to replicate itself for new virus to proceed. And so when you have a life cycle of a virus, the important thing is the cell permissive? If you have a permissive cell, it will allow an infection to take place and virus particle to form and produce. It's also, there are some circumstances where you'll have a permissive cell, but the DNA can actually either live episomally or actually integrated into the DNA and be quiescent. So you can have active productive infections or latent infections. However, there are some cell types, some hosts that are non-permissive, where the protein does not let natural virion capsules be formed, or it will actually, in some cases, they get in the cell and will actually transform the cell. And so you don't really get the infection that allows the virus to replicate itself, but it could actually be a cancer transforming event. So these are events that occur every day when we talk about viruses. We forget this basic biology. So it does turn out, we know now that, I put this picture, I've shown this picture before, graft versus host disease is T cells gone wild. This is a sloughed gut. This is GI tract where the entire mucosa is denuded. And, you know, if the person is going to survive, they have to re-epithelialize the entire surface. Well, we've now learned that we can actually harness this destructive power of of T cells into a therapeutic, which is CAR T cells. And we now use CAR T cells frequently and across the world. And again, I tell people, you know, in 2018, when our lymphoma CAR T was approved by the FDA, I really didn't think many people were going to use it because it was going to be, the cost was so expensive. Well, at the ASH meetings, 4 months ago, they announced that over 35,000 people across the world have now received CAR T for either myeloma or lymphoma. So CAR T has really demonstrated that we can genetically alter T cells and harness the power of the T cell. So what about viruses? So this is a picture of a paper that we wrote 10 years ago or so, which was a really interesting paper where we summarized our adenovirus experience. We have had, at the time in 2010, we had, I think, 6 or 7 systemic adenovirus events of patients, only 1 survivor. But this was a patient, you could see his liver functions were doing fine, and all of a sudden, in a matter of days, he was in fulminant liver failure. And was this GVHD? Was this, you know, what was going on? Well, we got a liver biopsy. You could see that's a transaminases in the close to 10,000 versus where it was normally in like 60. And it went up in a matter of 72 to 96 hours. We did get a liver biopsy. This is the liver. There are there's no visible architecture. The entire liver was necrotic. And then when we actually looked at it at high power, you could see that what's left of the hepatocytes, they're ballooning, they're becoming apoptotic, but they all have inclusion bodies in them, which was a viral inclusion body. When you did immunohistochemistry, this was positive for adenovirus. And then we actually sent it off for, which was instructive, we sent it for electron microscopy. And this is a single nucleus. But you can look at all the virus, all these are independent virus capsids that are ready to lyse, be spread into the tissue, and then infect other cells. So a single cell infected is given hundreds of new viral particles. And so you can imagine if you can actually, 1, you can see why that would necrose a liver, but 2, if you could capture that power, maybe it could be done therapeutically. So again, a slide I modified from the past is, you know, we've said that the standard cancer therapy has always been surgery, radiation, and chemo. Immunotherapy and cellular therapy has become a major, major player. And I'm suggesting that maybe oncologic viruses are going to become an entity. And it's mostly going to be focused on, just not the destructive power, but where it's going to be under the category of immunotherapy. So what about the immune system? To remind you aspects of the immune system, we have 2 major arms. We have the adaptive immune system, which has to be instructed. That's B cells and T cells. And we have the innate immune system, which is the neutrophils and the monocytes and macrophages that respond immediately. Somewhere in between is natural killer cells. NK cells have some innate activity, but there's some evidence of NK memory, which would mean put it more in the adaptive immune system. And it's important to recognize in cancer biology and also in infectious disease biology, they are coordinately active. And how does it work? When a bacteria, you know, gets into you, you have lipopolysaccharide on gram-negative rods. You have a host of viral molecules that, you know, that can activate the immune system. And there's also, when you have cellular necrosis, you have endogenous cellular material from inside the cell that could actually lead to activation of the immune system. And these are called PAMPs, pathogen-associated molecular patterns. And what the PAMPs will do is they will activate the toll-like receptor. Toll-like receptors are critical not just for bacterial infections or viral infections, but it's sterile inflammation. So cholesterol crystals, asbestosis, it all works by toll-like receptor activation to create an inflammatory signal. But this whole cascade of the toll-like receptors is really critical to activation of the innate immune system to try and protect us from infection. I will also point out, again, that there is the adaptive immune system where we have lymphocytes, B cells, and NK cells. We have these other nonspecific elements. But critical also to viral immunology is that we have HLA molecules. We have HLA class 1, class 2, similarly looks the same. And these are pictures from the 1987 original crystal structure paper. This is the schematic, but this was looking down at the helix, the alpha 1, alpha 2, and how it creates a pocket. Well, when they originally crystallized it, in blue is the actual amino acid structure that predicted the crystal structure, but they noticed this little debris in the middle. Well, they didn't really, initially in 1987, they didn't pay attention to that, but what they ultimately showed is that is Epstein-Barr virus peptide, because the original crystallization came from the JY HLA-A2B7 homozygote Burkitt lymphoma line, EBV transformed line. But this little stuff that they noted, it turned out that was Epstein-Barr virus peptide that was captured in the pocket during the crystallization. So how do viruses get in? So it turns out that viruses, through those little, whether it's the spikes or those coat proteins, they have identified eukaryotic targets that will help them get in. Some of them are sugar, carbohydrate side chains, sialic acid, terminal sialic acid is a receptor for some viruses, a series of cell adhesion molecules, CD4 obviously for HIV. ICAM, remember CD54? It turns out it's the rhinovirus receptor. It turns out there are some tyrosine kinases or phosphatidylserine receptors that viruses can utilize to get in the cell. And if we even go further, we've identified some of the viruses, like here's adenovirus, can use, cell adhesion receptors and integrins to get into the cell. Zika virus uses the acyl tyrosine kinase to enter the cell. Sometimes it can work by endocytosis and to be pulled in, and sometimes it can actually direct itself to get in the cell. And once it's in there, it will activate the cellular machinery. Now, a little bit of history. When did people first get a clue that viruses could interact with cancer? Well, going back 100 years ago, this is some from old, some old reviews. There's case reports in 1904 of a woman identified with acute leukemia. She acutely got sick. That doesn't surprise us. We know that patients get sick with leukemia. This particular person went into complete remission. 10 years later, there was a report of a woman with cervical cancer who again got sick. And then while she was sick with what was assumed to be a viral illness, again, they didn't know much about electron micrographs. They didn't know about viruses, but she had massive tumor necrosis of the cervical cancer. And over the years, there was these anecdotal reports in the literature about measles, influenza, rabies, chickenpox, all being all influencing and causing tumor regression. In the 1950s and 60s, people actually started clinical trials using viruses, and these were some of the candidate viruses that they used to infect people. And the proposed mechanisms in retrospect was, well, when you infect a cell, the virus is going to turn on higher levels in the cancer. It's going to take over the cell machinery, but you're going to turn on tumor antigens. You're also going to activate those danger signals, the DAMPs now, and that'll break down a tumor immune tolerance and will let the immune system go into an area where they couldn't get in before. The viral particles also, as they start to break out and create inflammation, it's going to stimulate now what we know as chemokines that'll stimulate the immune system. We do know, though, viruses have a tropism to tissues of interest. So again, rhinovirus gets the epithelial. There's a CD4 or CD54 or ICAM on the respiratory epithelial mucosa helps, rhinovirus get in. And a number of years ago, I haven't really checked on what happened with this. There was a company that was making a nasal inhaler of soluble CD54. So the moment you started to get symptoms or maybe you were exposed, you could just do a nasal puff like you do Flonase, and you get soluble CD54 to block up the rhinovirus. Honestly, I think the cost of the protein, it was was prohibitive to actually treat rhinovirus, but it was an interesting thought. The important thing, though, what a lot of people feel is we all now talk about the cold tumor that's immunologically inert, and that what we're doing is make it inflamed, and that may be part of the treatment. Well, I actually did go back and look, and in 1952, I thought this was an interesting paper published in the Annals of the New York Academy of Sciences. And so this is a person at Sloan Kettering. They were developing their cancer biology, and what they did is they had animal models for sarcoma. And so this is a animal model, where they do a tumor implant. On the right is healthy-looking sarcoma. This is in mouse. And now, if you look on the left, that's the same sarcoma, but this was infused with a Russian encephalitis virus. And you could see that the tumor completely necrosed. And what they ended up doing is they went through a series of viruses, and they asked the question of what is effective against animal tumors and what viruses do they not seem to get efficacy. But you're starting to see that people are identifying that there are viruses that does seem to be associated with the ability to break down a tumor. So people are interested in this this is 75 years ago. But it's taken a long time to get to where we are now. So what do we know? So now general mechanisms, what do we think's happening? A virus will infect a cellular target. The virus gets in because of PAMPs. It activates toll-like receptors. Toll-like receptors turn on tissue-based dendritic cells and other antigen-presenting cells. They start to stimulate cytokines, including tumor necrosis factor and interferon, that will then lead to interferon lead to upregulation of MHC antigens, which will amplify the immune response against the infected tumor. The interleukins will, with TNF and interferon, will drive a helper 1 response. You'll also stimulate natural killer cells, but what you do is you cascade a response leading to cell death, and then you lead to the PAMPs are the pathogen associated, but now we have our own innate stimulators are called DAMPs, or damage associated molecular patterns. That's the intracellular uric acid getting into the extracellular medium. DNA getting into the extracellular medium will actually trigger and amplify the immune response. This was interesting. It's reported that in solid cancers, 65 to 70%, with all the genetic mutations, they actually have defective interferon signaling. And so that's when if you give a virus, they are going to potentially have a distinct response from normal cells, which has normal interferon signaling. And so you may be able to capitalize on that selective, a differentiating characteristic to attack the tumor cell. And so when you want to reproduce it, so the fact is, though, if we are going to try and do this, we got to make it safe. So we have to inactivate virulence genes, and then we have to figure out how we're going to deliver it. Is it going to be local or systemic? And so this is a paper that came out last year on this issue, is you got to remember when a virus comes in, it is going to stimulate the immune system, and so you can get an immune attack to clear virus. That's what immune system, the evolution of the T cell immunity and B cell immunity is to protect us from virus. Cancers are just a mistake of time, and so, but if you want to ultimately get your virus in to maximize an anti-tumor effect, you have to focus more on letting the virus turn on tumor antigens, activate those innate immune systems, maybe, stimulate anti-tumor T cells and avoid antiviral T cells. But ultimately, there's going to be a balance of the immune response to the virus versus the immune response to the cancer if you're going to use and try to capitalize on the activity. I borrowed this picture. This is another from another review from last year. Here is a host of viruses that are all being tested to be oncolytic and how they could be oncolytic. It turns out that how do cells die? Well, there's multiple cellular biologies associated with cell death. You know, apoptosis, we talk about them most. Autophagy is one, necroptosis is another. But it does turn out that individual viruses, and I put it in yellow, are more likely to cause one type of cell death than the other. So we have to understand these biologic pathways if we're going to really master, you know, the use of oncolytic viruses. So as far as viral families, what has been under consideration? Well, these are the current major oncolytic virus families that people have been focusing on their research over the last 30 years. And most of them are double-stranded DNA viruses. Some of them, like the pox viridae, picornavirus is an RNA virus, but most of them are double-stranded DNAs. And just to talk about a couple of these viruses, adenovirus doesn't have an envelope, but it is double-stranded. There are 7 species of adenovirus only that infect human cells. Because it's big, it's 36 kb, you can actually have room to put in some transgenes. So adenovirus is a good thing to carry you can use it as a carrier into the eukaryotic cell that you want to attack. It will not integrate into DNA because it only replicates episomally. So it doesn't go into the DNA. And so adenovirus serotype 5 species C is the one that's been most studied. And there's been multiple safety modifications that people have made with different strains. And I'll point out that the one that the first one used was treated a head and neck cancer and nasopharyngeal cancer in China. They deleted the gene E1B. And what that does is, remember, P53 is often lost in tumors. And so if you take the gene E1B out of adenovirus, it actually potentiates selective infection of that attenuated strain only into cells with defective P53. Cells that have normal P53 will be resistant. They've actually taken this particular strain to a phase 3 study of chemo plus virus versus chemo alone. Increased overall response rate was proven in nasopharyngeal carcinoma and head and neck. And this is officially approved in China as a treatment option. However, they do note that over time, neutralizing antibodies will limit the benefit. And so they continue to genetically modify viral strains. They do want to emphasize, though, that monotherapy, virus alone, doesn't seem to do the trick to treat advanced cancer. It seems to work in combination. HSV1, I want to also highlight, it's an envelope virus. It's a double-stranded virus. Another large genome, it's 152 kilobases. And there's 30 kilobases what's felt to be non-essential genes for infection. So if you get rid of those, you can use that. You can use the HSV as a packaging vector. When you use HSV, there's not been shown to be any insertional mutagenesis. It's not like, Maloney leukemia virus. It does replicate in the nucleus, not in the cytoplasm like adenovirus. You can introduce a safety signal. The first work that was done was actually using a modified TK, using the TK gene to infect cells, but they put a safety signal in there that you could just kill the cell. So, like you could have, like I said, you could have a target cell receptor. So if your HSV infects the cell, it will also turn on target cell like HER2 or EGFR. And you could use an antibody. If you get herpes virus infection out of control, you could turn it off with an antibody. What the major modification is, they use the gene for GM-CSF, which will also, remember, GM-CSF will stimulate your monocytes and macrophages and antigen-presenting cells. They have shown that this can be done safely. It's been used in these diseases. And again, it's been tested as monotherapy in combination with checkpoint inhibitors. Paramyxovirus, I mentioned again just because that's mumps and measles. It binds to like CD46 or SLAM, which we know is a target for in myeloma. What's interesting, I'll say an anecdote, is that there's a patient from that was reported. There's a study that was done at Mayo Clinic around 2010 where they were testing measles virus, and they had a myeloma patient, had 2 transplants, ultimately end-stage refractory disease, and they treated him with measles virus and a attenuated strain. And just with the virus alone, the person went into a complete remission and was in remission at 5 years after report. So again, it's an anecdote. Everything's an anecdote in this business, but it gives us opportunity. Finally, there's the pox virus. And I just mentioned this one because this is smallpox, cowpox, and also vaccinia. And we understand these receptors. They're very, very immunogenic. They also are easily armed with immune enhancers. But there's a very important clinical trial was done where it was looking like this was going to be a treatment for liver cancer. And so Pexavec, it actually went to phase 3, where they had a tyrosine kinase deleted, GM-CSF armed virus plus sorafenib versus sorafenib alone. It turned out no benefit. So it worked really well in vitro. It seemed to have a good positive signal in phase 1, phase 2, but overall, in an intent to treat, no significant benefit. I would say, though, it is interesting. There are studies now going in melanoma right now where they're arming using vaccinia as a vector, but they're turning on CD54, CD82, and LFA3, CD58, which should really maximally stimulate the immune system. So you could give this virus, stimulate the immune system, and hopefully turn on a cold tumor into a warm tumor. So what does it take to make this work? Well, you have to pick the right virus. Like I said, size plays a role. Large viruses can be, like I said, manipulated for genetic payload, but it's really hard to get it into tissues. And small viruses can really get into tissues quickly, spread across a tumor, but you don't have the ability to genetically modify. So you have to attenuate it to make it to be the virus you want. RNA viruses replicate in the cytoplasm. DNA viruses are more likely to get into the nucleus. Again, aspects of biology you have to understand if you're going to actually develop this. And what people are now focusing on is, like in some cases, can you make a packaging envelope? Can you mutate the envelope so the envelope is very, very antigenic to the immune system, but turn off oncogenesis so the virus doesn't transform? Delivery is important. You have to understand how do you get it in. Like people are working with glioblastoma, they're doing direct injection into glioblastoma. So that does seem to be very safe if you give it directly into the tumor, but you have to be able to get to it. Systemically, there's no guarantee of how you understand how the virus traffics, but it does seem that you do need to have repeated dosing. Immunologic clearance is an issue with systemic delivery, and now people are developing new delivery systems, nanoparticles, liposomes, a whole host of things that would allow it to get in, as well as decoy viruses that will, you give the decoy virus first, so you eliminate the neutralizing antibody, then you come in with the therapeutic virus. And as far as their strategies, again, a take-home message is monotherapy in general fails. Combination therapy seems to be the way of the future. These are the malignancy targets that people look at: melanoma, bladder, CA. These are the ones that people are concentrating on. And so this is important. Where is this in today? I mean, honestly, I did not know this. These are the 5 viruses that are officially approved by regulatory bodies across the world. This is as of 2023. There's only 5, but they are in use. And this goes back to the adenovirus for nasopharyngeal I mentioned from 2005, and most recently is nadofaragene firadenovec for bladder cancer, non-muscle invasive, in 2022. And I'm going to talk about just a couple of these. So what about the FDA-approved therapies? The first is telamogene or TVEC. This is a herpes virus engineered virus. It's also been attenuated, so it has live replication in tumor cells. It will induce immune response. Importantly, remember, herpes viruses can get into nerves and live. Like you get cold sores, it's in your nerves, it just reactivates. Well, they eliminated they deleted the neurovirulence gene. They also, there's a gene that actually turns, that the HSV virus normally does, that will block the Golgi apparatus loading a peptide into MHC molecules. And so they deleted that gene. So now the HSV virus can actually, the peptide breakdown can get into MHC and be immunogenic. They also turned in and expressed GM-CSF, again, to turn on macrophages and monocytes. It is sensitive to it is acyclovir. I mean, it's a herpes virus. That particular strain is still sensitive to acyclovir, ganciclovir, any of those agents. It turns out a lot of people do this for direct injection. This is a big virus. This is not something to give systemically. And they've done a phase 1 study with multiple cancers. They showed it can be given in injections, by injection, and it was done safety to some local injection site reactions. They've seen in the first study published in 2006 of TVEC that there were 14 of 19 patients had some evidence of tumor necrosis. They then took it to phase 2 in melanoma only, unresectable stage 3C4, 50 patients. Overall response rate was only in a quarter of the patients, but 10 complete remissions. And of those patients who had response, duration of remissions was as late as 40 months. And it was all based on localized immune responses. So it could not, they didn't demonstrate a systemic immune responses, but local responses. So this led to the phase 3 Optimum study, 436 patients, unresectable, locally advanced melanoma. This was mostly not surgically advanced, or not mostly systemically, metastatic, but locally advanced. It was 2-to-1 randomization of TVEC versus GM-CSF alone. TVEC, you get virus plus GM versus GM alone. And basically, it's a year's worth of treatment. There's 24 doses of viral product. Basically, you loading dose for 1 month, and then every 2 weeks you get a dose, and versus 26 doses of GM-CSF. And if you were responding, you go for 6 additional months. Well, what they showed was there was definitely durable responses. TVEC, 16% versus best available therapy of 2%. Overall response was 26% versus 6%. And there were 11% CR rates. Importantly, there was a trend toward better overall survival. And interestingly, when you start treatment, it takes time to get an immune response. So almost half the people looked like they were progressing before the disease regressed. And so you can't, when you commit somebody to this, even though it looks bad, you keep going. But this led to FDA approval over 6 years ago. So I'll just show you a waterfall plot, basically. Here's the patients who gained best overall response. Here's the patients with GM alone. And versus the absolutely progressed. So clearly, there was a far superior outcome with the TVEC product. This was published in 2019, the final results of the data. And you can look at this plot, also see that what favors TVEC is a host of things that you can overcome advanced stage disease, poor performance status. There's a lot of advantages, and the overall survival is improved. But just to show you our own experience, I contacted John Beto, who's our surgical oncologist, and we've treated 13 patients with TVEC over the last 5 years, since 2019. And so he sent me a patient. I mean, he just gave me the patient. I made this case In May of 2022, you had a patient who had a nodular melanoma, and it was a stage 3B. It was resected. It turned out it had a mutation of BRAF, but the mutation would make it not sensitive to the various TKIs. The patient said, I don't want to take adjuvant therapy. They were recommended to get adjuvant therapy, but didn't want to take adjuvant checkpoint inhibitor therapy because he had pre-existing pulmonary fibrosis. He was afraid of getting lung injury. So he said, I'm just going to be observation. Well, within a half a year, they found evidence of progression. So they set him up and decided they're going to start with TVEC, and they treated him for 17 cycles over a one-year period. And so in March of 2023, this is what his arm looked like. And this is basically over every couple of months. He took pictures. So this is March. This is June. It sure looks like it's worse. Remember, 50% of the patients have disease progression before you get a benefit. It does look thicker, but you notice the margins aren't extending. It's still staying localized. And then by September, it seems to be regressing. This is November, and now March of 2024, it's macular. They biopsied it. There is no melanoma cells there. What this is residual melanin pigment left in the tissue. And so at this point, after a year of treatment, they're saying they didn't recommend the 6 more treatments. They're just going to hold. And this is all local injection. So here's a case where OHSU has put somebody into complete remission. I'd say that there's guidelines. This is in your record. If you do ever use TVEC, there's specific guidelines of how you should use it and how you can manage it effectively for your patients. This is the other drug that's approved. This is the only other gene therapy. This is the only other viral therapy approved, and it's a modified adenovirus. The other's a herpes virus. It's non-oncolytic. It cannot replicate itself. And what it is, it's for topical therapy. If you have topical bladder cancer, a lot of people will get BCG to stimulate immune response. So it's non-muscle invasive bladder cancer. But if you now have BCG failure, you can now put this virus in, and the virus is genetically modified to turn on, to express and turn on interferon alpha. You directly instill it in the bladder. You let it sit in the bladder and just coat, and the cells, the adenovirus will infect the cell and will actually deliver it into the topical urothelial cells. This went to a phase 3 single-arm, multi-center study. They've identified a certain dose of particles per milliliter. You infuse the bladder with 75 milliliters, and you repeat dose every 3 months. And what they showed is a 53% complete remission in 3 months, median duration of response of 10 months, and overall duration, if you're a responder, was almost 50% at 1 year. Now, it turns out this was approved 14 months ago. Nobody at OHSU, I talked to our urologist, no one at OHSU has been treated with this because at the moment, the current guidelines are saying if you're BCG resistant, the current guidelines are do a cystectomy. If you chose not to do cystectomy, you can do checkpoint inhibitors with a 51% CR rate. Here's a 53% CR rate. Checkpoint inhibitors are systemic with a risk of systemic toxicity. This is only local. And the median duration. Now, this is interesting. Just last week, interleukin 15, a mutant interleukin 15 that's fused with the alpha chain, which binds to only the high IL-15 receptors on both NK cells and T cells, has now been approved for the exact same thing of non-muscle invasive bladder cancer. So in this very relatively rare circumstance of BCG unresponsive topical disease, we now have multiple therapies, including virus, cytokines, checkpoint inhibitors, surgery, and chemotherapy, all as options for our urologists to choose. And this is showing what the gene therapy does. Yeah, not everybody responds. This was patients who had carcinoma in situ. These were high-grade, locally invasive, and you could see good responses, though, for the topical viral therapy. So oncolytic virotherapy works. Well, we always have to know that if we're going to do something, we have to know what we're doing. Like everything else, it's a drug. So we have to be aware of safety considerations. We have to monitor when we do any virus for local or systemic inflammatory response. We have to be aware of, if we do systemic, that there can be hepatic or hematologic toxicities. There can be viral shedding. That's always a worrisome. You're giving something attenuated virus, but you may have somebody in the family or someone in close contact, especially an immune suppressed person or it's a pregnant female, may be at risk for those infection from those attenuated strains. And currently, people are doing experiments making chimeric viruses. They're making RNA, DNA hybrids. They're creating larger packaging machinery, by creating viral fusion, whatever they're hybrids between the viruses. Now that may sound like too much genetic engineering and dangerous, but that's what's happening. These are practical considerations if you're going to manage oncolytic viruses for using how we use it, either direct injection or systemic. And just mentioning that, I said combination is the future, and people have definitely done it with chemotherapy. They've definitely done it with radiation. They've definitely done it with immune checkpoints. And I'll show you that people are now looking at targeted therapy in CAR T cells. I'm it's getting to the end of the hour. I'll just tell you that this is a study of combined virus and radiation therapy. The key is down here. If you look at TVEC with radiation therapy versus control or just TVEC alone or radiation therapy alone, this is injectable tumor into animals, and you can see that you get a better, with combination, you get better tumor control over time. And it does seem that, the animal survival was improved by combination therapy. These are just variations of the experiment. And here's a case of squamous cell carcinoma that got an oncolytic virus with nivolumab and radiation, and you really could see a very rapid response over time. This has taken over years. It wasn't just local because they got local benefit, but over time, there was a small metastasis, but there seemed to be a systemic response when they actually looked 5 years later. This person's in continuous remission. He only got treatment for about less than a year, but he's now 5 years out in continuous remission. So again, maybe it's a tool for the future. And for my interest, these are now doing animal models of oncolytic viruses in combination with CAR T. So this is for solid cancer especially, because CAR T for solid cancer is difficult, but they're using the oncolytic virus to turn a cold tumor hot, and then once it's hot, it should invite the CAR T in, and hopefully the combination therapy will lead to a new vehicle for treatment of the solid tumor. And so this is just showing, this is a paper that's actually showing in mice that they're able to get better responses of a tumor with CAR T, if you actually miss it with an oncolytic virus first. And so in conclusion, this has been an ongoing tempting therapeutic option. There are many currently developed replication incompetent viral vectors that are being used for gene therapy, like for CAR T, we're using, lentivirus to get the virus in. Well, now people can do the same to use therapy for, actually to use not the virus as a gene therapy vector, but use the virus as a therapeutic vector. Monotherapy seems not to be as effective, but we want to do genetic modification will lead to safer viruses. That's what led to FDA and other regulatory approval. But I do believe that combination therapy, from what I see now and where I envision the next 5 years are going to go, I think the combination therapies are going to emerge as options that we will be using. So, as I say, this was me deciding to learn something new and share something with you. This literally is something I realized I didn't know much about. It's happening around me, and I said I should learn a little. And so I thought I would share, and it's only a snapshot. It's been ongoing for 100 years, but stuff keeps coming. And I just thought I would let you know, and I hope this was of interest to you. And as we said, every once in a while, you guys can ask us, but this would, if this is going to be a, this is the, as I was told by Wanapa, as that these are used for CME, and hopefully this is an educational session that gives you some benefit that you enjoy and that you can think about for the future and share with your own team members out there.
Thank you,
Thank
Prof. Very, very comprehensive learning for me today. Dr. Surapon, any comment from you, please?
So I think it's a very inspiring lecture. So, Richard, I would like to ask you whether this kind of treatment may be beneficial for hematologic malignancies.
Well, yeah, I think
Genetically.
no, I think that's a great comment, and I think it's for hematologic malignancies. As I said, the measles virus sees the SLAM antigen. Remember, that's what there's an antibody therapy. The elotuzumab, is a SLAM 1 target. So yeah, there's myeloma would be a target. There's some would be, I think in lymphoma, we know that there's some of the target antigens are activated that could also be targets. So I think for lymphoma and myeloma, those are the low-hanging fruits for hematology. But honestly, we have so many other tools that there's not a lot of attention going on for viruses or viral therapy. But I do think it can be done. And like I pointed out, that patient from the Mayo Clinic that got treated with the measles virus had a 5-year remission.
Are there any studies going on breast cancer?
I don't know that. There was definitely there's some viral studies with breast cancer. I don't know of any personally. I haven't looked for that. I just, all the focus obviously has been on melanoma and, and the head and neck cancer from work in China. But there were studies, using, targeting metastatic breast cancer as an option. I'd have to go back and look. And, I think in the United States, MD Anderson and Sloan Kettering have been very involved in these type of, developing oncolytic virus studies. So that's something we could check into.
Good. Thank you. Thank you.
Any other question from other side? Ajanutra, any question from you?
I didn't have a question, but your lecture from Richard is very, very inspire us, and I think in the future maybe it will be expand to the pediatric